Hey everyone, Mr. Murphy here. I'm going to pick up with chapter 3.1, and in this chapter we are looking at nucleic acids, proteins, and enzymes, beginning with nucleic acids. So this will be pretty quick. I've mentioned in class that nucleic acids are not exactly the most important component of this unit. However, being one of the four major classes of macromolecules, it is important to understand how they do follow the same rules as the other three, and we'll circle back to it in later units when we get to genetics. This chapter in general, though, follows the following path. We go nucleic acids, proteins, enzymes, and then we look at regulation of various metabolic processes with enzymes, so ways in which we can sort of turn them on and off or just regulate their activity. So the chapter opens with a question of how does understanding proteins and enzymes actually help to understand how aspirin works, and in the last video of this chapter we'll circle back to that. So nucleic acids, like I said, it's not the most important thing right now, but we do want to be aware of them, which they are another macromolecule formed primarily by carbon, hydrogen, oxygen, nitrogen, and phosphorus. And basically the two main types of polymer is DNA and RNA. The monomer are nucleotides, which are composed of a nitrogen base, a sugar molecule, a 5-carbon pentose ribose sugar, and a phosphate group. And these get linked together and form the sugar phosphate backbone of DNA, and then the bases are the A, T, C, and G of DNA. There are two main classes of these, pyrimidines and purines, and we'll dig into the significance of that in a later unit. The sugars are different depending on whether we're looking at DNA or RNA. RNA also has a difference in terms of the fact that it has uracil instead of thymine. There is the linkage between these when they are undergoing dehydration synthesis, which is illustrated here. You see that an H here on the phosphate group and the OH hydroxyl group on this sugar are responsible for forming that water molecule that's pulled out. The oxygen here then links up to the carbon here that's missing a bond, exactly what we've seen before. We call this a phosphodiester bond. So just like glycosidic linkage or ester bonds in the case of lipids or peptide bonds in proteins, it's just a name for the specific type of dehydration synthesis or condensation reaction that's occurring here. So we have a whole host of details about the differences between these different types of monomers and polymers and the significance of their arrangement and things like that. We'll save that for later on. I would say it's just good biological knowledge to understand some broad differences between RNA and DNA in terms of what the bases are. RNA is also a single strand, where DNA is a double strand, that double helix, and then the sugar is different as well. This is another great example of hydrogen bonds. So the pairing of adenine and thymine, A and T, and the pairing of cytosine and guanine, it's consistent, and you don't see A's bonding or pairing with C or G because of hydrogen bonds. So we'll talk more about this with proteins, but the arrangement of these molecules with respect to each other, if we have a cytosine here and a guanine here, it's important that they're anti-parallel, they're opposite to each other. Not something we need to worry about until we get into genetics, but the way they line up together, their hydrogen bonds actually align in a reliable way. And so that's why thymine and adenine, they're kind of mirrored to each other with their available hydrogen bond attractions that can occur between these two different sites and three different sites here in cytosine and guanine. So it's a great example of hydrogen bonding. So that's an important point to recognize, if we were to draw questions down the road or on the AP test that are pointing to nucleic acids as sort of a source of a way to talk about hydrogen bonds. It's what holds DNA together. It's just many, many hydrogen bonds. You saw 2 or 3 hydrogen bonds depending on what the pairs are. And if you have millions of nucleotides linked up together, you can see why that would add up to a pretty strong molecule, despite the fact that individual hydrogen bonds are very weak. So RNA is single-stranded. It still folds up into sort of unique shapes, and here's an example of that. So even though RNA is single-stranded, like or complementary regions of that RNA will still line up together, and so they can kind of fold up, very similar to what we'll see on the next section of this chapter with proteins. And so getting ahead of ourselves again, there's a lot of fascinating evidence that would support the idea that perhaps RNA predates all of life, and that it was early RNA molecules that were sort of spontaneously generated and able to line up together and form these interesting and functional three-dimensional shapes that may have been the early precursors to life. So significance to the fact that they do this hydrogen bonding and sort of folding and things similar to proteins. But again, that's more for another day. So DNA, meanwhile, forms that double helix. It has this double-stranded side where you have these complementary bases on each side for the entire length of the DNA molecule, and that's what forms our standard DNA. So DNA is an informational thing, so is RNA in terms of storing genetic information, and then the processes of transcription and translation, things that we'll get into later on, allow for basically the extraction of that information and the use of it within a biological organism. So I'm not going to go into any sort of transcription, translation. That is not stuff that we need to review right now. But again, it's always helpful to sort of have these things in the back of our minds as we're talking about other concepts within biochemistry or cell processes, so that we can be sort of seeing the connections and how all of this stuff is ruled by the same fundamental rules of chemistry. But again, that's not something specific for this unit. Some more terms on DNA and some more information on all of that. That's the end of 3.1. So a quick look at nucleic acids. It's a good vehicle for asking questions about the more broad biochemistry stuff, but we will return to this in great depth in a later unit. So if you have questions, let me know.